I'm Randy. I'm one of the curators, and my pet interest is the quality of the data. So if anyone finds errors and submits them to us, I personally will take a look at them. So this is where we are in the day. After me, Carrie Nima will talk about how to use the search interface. And at the end, Bjorn will give an introduction to the analysis resource. Some of this may be what Bjorn mentioned, but I'll go into a little more detail. So the scope of the IEDB, as he said, was infectious disease, allergy, autoimmunity, and transplant. But HIV and cancer are excluded. I know a number of people said they were interested in cancer. We do have some cancer epitopes. We capture HIV and cancer epitopes when they occur in a manuscript alongside infectious disease, allergy, autoimmunity, or transplant epitopes. And we also capture those epitopes when there's a PDB structure demonstrating a TCR, antibody, or MHC structure with an epitope. So all the data in the IEDB is experimentally confirmed. We do not capture predictions or reviews. And we do include negative data and supplemental data. So each of the experiments in the IEDB would demonstrate the binding of a T cell, an antibody or B cell, an MHC binding experiment, or MHC ligand elution experiment, where that receptor is epitope specific. So if a known epitope specific receptor was tested in other experiments other than binding to the epitope, we actually also include those. So if an epitope specific antibody neutralizes a virus, we would have that experiment as well. So our criteria for including epitope data, if it's a linear peptide, it needs to be 50 amino acids or less, and it should have been tested as an immunogen or antigen to be demonstrated in the epitope. Or if it's a discontinuous epitope, those residues should have been shown to be important in recognition. For example, by point mutation of the residues of a protein, you abolish antibody binding, that demonstrates it as an epitope for that antibody. We also capture nonpeptic epitopes, carbohydrates, lipids, drugs, chemicals, and those need to be 5,000 Daltons or less. And as was the question that was raised by Evan, and Bjorn mentioned, we need a minimal information to include these experiments. So the epitope itself, we need to know the structure. We need to know the linear amino acid sequence of the peptide, or if it's non-peptidic, the authors needed to have provided what the structure was. And we need to know the host, what organism did the T cells or antibodies come from. And we need to know the outcome. If we can't determine if the experiment was positive or negative, we can't add it to the database. And we do write authors regularly to get all of this information, because our goal is to include as many manuscripts as possible. So we do not create the data. We get the experimentally generated data from the public, most of it the published literature, and some of it from the submissions, as Bjorn mentioned. So the literature curation process, we began with PubMed. We do a complex query of PubMed every 2 weeks, searching for new epitope-related literature. And as we said, we've curated the majority of this. I've been told we have more than 99% of the epitope data in the database now. So we then classify all of the references we retrieve from PubMed using an automated classifier that determines, are they within our scope and are they likely to be curatable? And that means they have enough data to be put into the IEDB. So we classify the epitopes by category, and then we give them to the curation team. And the curation team, they read the curation, enter all of the experiments into the database, and then another curator will read the same manuscript and enter all of the data, review the data previously entered. And they work together to complete all of the fields that we capture. And once a week, new data is pushed to the external website and becomes publicly available. And that is about 15 to 20 new publications per week. So to make sure that the data is entered consistently and accurately between each new publication and among each member of our team, we have a PhD level team of 8 scientists at La Jolla Institute and 1 at KEBI and EBI, who handles our non-peptidic epitopes. And all of the data is entered using our formal curation guidelines. We have a curation manual that's publicly available, and you can get to it through the website. And it explains how we curate. We have a lot of rules to handle every scenario we encounter in the literature. And then the peer review system, as I mentioned, to help assure accuracy. We also use external immunological experts. We refer to these for how the design of the website in the first place. The design of new fields, if we encounter something novel in literature, if new scenarios come up, we don't just decide on our own in a bubble, and also the meta-analyses help reach out to experts. And so the application that the curators use themselves has its own built-in validation. So as they enter one field of the database, another field will be required or disallowed, or if they pick a select term for one field, a subset of terms will be allowed in another field. So this helps ensure consistency. And the submission community that Bjorn mentioned is primarily the epitope discovery contact holders, and these are some of the infectious diseases and also allergens that data relates to, and these are some of the main submitters who have those contracts. And the submission process is similar to the curation process, except that the submitter submits the data to us rather than our curation team taking it from the literature. And when they do the submissions, they can use two processes where they can use a wizard that walks you through the process and it explains each field and the variable terms for those fields, or they can use a DST tool that uses XML. And usually when someone first submits, they use the wizard, and then once they're used to the database or they have a very large set, they use the wizard. And the same validation that is put upon our curated curations is also put on the submissions. So Nima is the curator who works with the submissions. He will work with the submitter and approve and make sure that each file is complete and consistent and makes sense and there aren't any missing values. And once they agree that it's complete, we will release the data to the external website, but we typically hold these until the submitter tells us when to release it, and that's because they often have a publication that's linked to it and they want to wait till the publication comes out, and then they release both side by side. And on the database, we link those identifiers so that you'll know that this submitted data is related to this publication. So as Bjorn mentioned, IEDB is a database of experiments. And these experiments originate in a reference. Oops. So each publication will describe one or more epitopes. And some publications will have several thousand epitopes. And each epitope must have been tested in one or more experiments. And each of these experiments is captured in the IEDB using up to 300 or more fields. And those fields describe the details of the experiment, from the host, the immunization, the antibody or the effector T cells, the antigen. And in order to control how we enter this data into the fields, rather than free text 300 fields, we use these standardized resources and external ontologies to help control how the data is entered. So for peptidic epitopes, we use the standard amino acid nomenclature, single capital letters. We do describe modifications if any of those residues are modified. And for each natural epitope, we assign it to a GenBank identifier. If the author gives us a specific GenBank identifier, we will use that, and we will let you know that the author specified that. If the author didn't specify a GenBank identifier, we pick a representative one that this peptide must 100% blast match to. So that's how we assign the protein source. We then group the protein sources using UniProt reference proteomes, so that within GenBank for any protein, there may be dozens of entries that are slightly different from each other, but they're essentially the same protein. We provide them to you grouped by the UniProt reference proteome. And for the organism source of that protein, we use the NCBI taxonomy. This is what Bjorn mentioned, that we slimmed down the NCBI tree to make it simpler and easier to use. We remove nodes that aren't relevant, and we use nomenclature that everyone's more familiar with. And for non-peptidic epitopes, we use a structure name given to us by the authors, and our CABI curator will read the manuscript, and they will enter the structure into CABI and give our curators the CABI identifier. And as these non-peptidic epitopes may be a smaller part of a larger structure, in this example, this is a small part of the LPS molecule that's recognized by an antibody. So the CABI curator will also give us that source, much like the GenBank identifier. This is the CABI identifier for that source. And the organism source for each epitope is defined by the authors. So, LPS may be present in many bacteria, but in this example, in the manuscript, the author would have said, this is the epitope of LPS from H. pylori. So again, we use NCBI taxonomy for that. And to describe the immunization process, how the host came to have the immune response. In the first place, what were they originally exposed to or injected with? It may be a mouse injected with a protein. It may be a human who has a disease. We describe that immunization process. If something was administered, we describe the route and the dose and the adjuvants. And if there were carriers, if it was a disease, we used the disease ontology to formalize the terms, to provide a hierarchy, and to provide us with link outs. And it also allows us to group similar diseases. For example, peanut allergy is a food allergy, so you may search on all food allergies, which you'll see later in the search portion. And then finally, the experiments themselves. We capture experiment types. Rather than us just making up experiment types every time we encounter them, we use the standardized nomenclature by OBI, which is Ontology of Biomedical Investigations. They provide us identifier for each experiment type, and they provide definitions, and we link out to them. And it also allows us to present the experiments in a tree form, so that you can see that if you want to search for a certain cytokine, you may not care which experiment was performed. You may just want all IL-2 experiments. We gain that by using that ontology. You also can see that it groups all cytokines together. And then if it's an antibody experiment, we describe the antibody in as much detail as the manuscript provides, whether it was purified antibody or serum that was tested, the isotype, whether it was monoclonal or polyclonal, if it has an antibody name, if it has an antibody proteins, if they provide GenBank identifiers for the chains of the antibody, we also capture those. And if it's a T-cell experiment, we describe the T-cells, how purified were they? Did they originate in blood or spleen or lymph nodes? Were they re-stimulated or not? If they were stimulated, we describe that. If they were direct ex vivo, we describe that. If it's a T-cell line with a name, we capture the name. And then if it's a T-cell experiment with a known restriction, either the authors stated or the authors determine it in that manuscript, we describe that restriction and we tell you how we know it. And we had to devise our own ontology for this, the MHC restriction ontology, where we modeled the protein chains that make up the MHC complex as genetic products of a genetic locus. And this allows you to search MHC information from a haplotype perspective, a serotype perspective, a class, or very specifically by each chain. And this also goes a little bit to the question that Evan raised, is that once we've put all this data in, now we've aggregated all the known experiments about all epitopes that are relevant to IEDB scope. And so you can see that one epitope is tested in multiple references, and that's the ideal goal of the IEDB, to put all that data together so you can see it built. And there are plenty of examples where one reference would find the epitope to be positive and another reference would find the same epitope to be negative. In some cases, this is due to differences, key differences in the experiment. And one is in a rabbit and one is in a human, or one was after someone had a disease and one was after an animal was injected with a peptide. But in other cases, they're identical experiments and they just have conflicting data. And the goal of the IEDB is to present to you all the data so that you can decide for yourself what is relevant to you and what is meaningful to you. And that's why the search criteria is so important. And then in other cases, the same epitope will repeatedly be shown to have the same results. And then that additive data will allow you to draw a conclusion that that's a consistently great T-cell epitope. This is similar to the same graph that Bjorn showed. It's that by putting all the data together, you can then make those decisions, and you can present this data as all B-cell or all T-cell or all mouse or all human, and you can see along the length of the protein which regions are consistently negative and which regions are consistently positive. And when they are positive, what's the response frequency of that positivity? So this region is positive in 25 to 30% of the subjects tested. So it gives you a weight to the data. And Carrie will go into great detail on how to use this feature. So now that we've accumulated all the data, you can see how much data we have. I made my slide in September. So you can see these were the total running totals of how much data we have. It changes every week, and the date of the update is always presented in the lower right-hand corner if you want to see if there's new data and how much. And then lastly, we're going to keep asking for your feedback. I think every speaker is going to mention it. But we do want to hear from you. It's very helpful for us. And every page, at the top of the page, there's a drop down for help. And at the bottom of every page, there are links to provide us feedback and help and make user requests. And as you go through the search interface today, you'll see frequently these little question marks. Those provide help. If you click on them, there'll be a very brief summary of what the term means. And if you click Learn More, you go to the Solutions Center, or there are full, long articles describing those search features and how we define our terms. And then also the database is free. We keep repeating that. It's free to search, and it's also free to export. You can export the entire database. If you go to the top right-hand corner, the More IEDB link drop-down gives you a database export menu, and it'll take you to a page where you can export the schema, you can export the database in XML, you can export the database in MySQL, and also CSV. And each page, when you're doing the searches today, you'll see at the top and the bottom of every page, there's a little Excel icon. And that means you can export your results from whatever query you just performed. Every tab of your search results is exportable in Excel.